Forensics laboratories in the U.S. use the CODIS system in their investigations. CODIS stands for Combined DNA Index System. CODIS focuses on 13 regions in the human genome, each of which contains a short tandem repeat, or STR. STRs are just one type of repetitive DNA found in the variable regions of the human genome. Early DNA forensics used restriction enzymes to cut the human genome and compare patterns of variable regions called VNTRs, or variable number of tandem repeats. This approach required obtaining a relatively intact sample of DNA that had not degraded. It also required a less efficient and more complicated laboratory procedure using the Southern blot technique. The CODIS system does not rely on restriction enzymes, but uses the polymerase chain reaction, PCR, to amplify the STR regions. On this slide, you see the TPOX GenBank sequence from the published human genome. It shows the short tandem repeat highlighted in gray, as well as the DNA code surrounding the STR. As you can see, the TPOX STR is an AATG repeat. In order to know someone has 11 repeats as shown, we don't sequence the genome and look at the region, which would be an expensive and time-consuming approach. We use PCR to make many copies of the DNA surrounding and including the STR, and this chunk of DNA amplified by PCR is called the amplicon. With 11 repeats, the amplicon is 126 base pairs long. The variability between individuals is a result of the differing number of repeats in a person's STR. For example, the individual shown might have 11 AATG repeats in the TPOX STR on their maternal chromosome 2. This individual might have 8 AATG repeats in the STR on their paternal chromosome 2. With 8 repeats, the amplicon is 12 bases shorter. 3 repeats of 4 bases yields a difference of 12 bases, or 114 base pairs long. Gel electrophoresis will separate these different-sized pieces into distinct bands. The banding pattern from different suspects can be compared to the banding pattern generated from the crime scene DNA. In lane F, the 11 repeat 126 base pair size band is above the 8 repeat 114 base pair size band. In gel electrophoresis, smaller pieces of DNA migrate through the gel more quickly and end up further from the origin after a set amount of time. So let's take a closer look at the reactants or ingredients in the PCR reaction and how PCR works. PCR takes place in a thermocycler that runs varying cycles of heating and cooling. Our objective is to make lots of copies of the STR regions. To make copies of a region of DNA, you need to provide template DNA. In a forensic investigation, one template is the DNA found at the crime scene. Suspects' DNA serves as the templates for additional PCR reactions. In addition, to make 1 billion copies of a specific STR region, you need to provide building blocks to build new DNA. Nucleotides are the building blocks of DNA. DNA polymerase is the enzyme necessary for synthesizing new DNA, and since the PCR reaction involves a series of repeated heating and cooling steps, the enzyme needs to be heat stable. The specific DNA polymerase used in PCR is called Taq polymerase. It comes from the bacteria Thermus aquaticus, which lives in hot springs and can withstand exposure to high temperatures. Finally, PCR requires primers, which are synthesized to be complementary to regions that bracket the STR. DNA polymerase requires a double-stranded priming site to bind and act. The primer binds to its complementary sequence upstream of the STR, which allows the Taq polymerase to act. Remember that 99% of even intergenic DNA is exactly the same in all individuals. This means that the primers are likely to bind in everyone. Primers are 25 to 30 bases long. Interestingly, a 24-base long stretch of DNA will be perfectly complementary to only one site in the entire human genome. Once the primer binds, the DNA polymerase, in combination with the available nucleotide building blocks, results in the amplification of the DNA region. The PCR animation that follows comes from the DNA Learning Center at Cold Spring Harbor Laboratory. It will guide you through the steps of PCR. In the first cycle, the temperature is raised to 95 degrees centigrade. This exposure to high heat denatures the double-stranded DNA, which is held together by relatively weak hydrogen bonds. The temperature is then lowered to 50 to 60 degrees, the temperature at which the primers anneal to their complementary sites on either side of the STR region. Next, the temperature is raised to 72 degrees, the temperature at which the Taq polymerase binds and extends the DNA one nucleotide at a time. So after the first cycle, we have two partially double-stranded pieces of DNA. What we are trying to obtain is two completely double-stranded pieces of DNA of our region of interest, that is, the STR region. This sequence of temperature changes is repeated over and over again. So let's go to the second cycle. High temperature denatures the DNA, the temperature is lowered, the primers anneal. The temperature is raised to 70 degrees, and the Taq polymerase extends the DNA. But we still don't have what we want, a double-stranded piece of DNA around the STR. In the third cycle, the steps are repeated: denature, anneal, and extend, and finally we have obtained two double-stranded copies of our target region of interest. These steps allow the number of target copies to expand exponentially with successive cycles. If we look at this in the graph, we see that after the first two cycles, we have copies of DNA, but it is not until after the third cycle that we have our first two copies of our target region. The number of target copies then becomes a greater and greater percentage of the total DNA in the reaction tube. At the end of 30 cycles, almost all of the DNA in the reaction tube is double-stranded copies of the region of interest, and in this example, only 60 copies of DNA are not target copies. When loaded on a gel, this amplified DNA will run as a single band that we can see with the naked eye. In summary, PCR is an automated process of amplifying DNA that requires small amounts of template DNA. In 2 hours, PCR generates over 1 billion copies of the target DNA.